For centuries, nobody knew why some people died after a blood transfusion while some others survived. In the year 1901, a scientist by the name of Karl Landsteiner, an Austrian biologist, physician and immunologist, identified the blood types A, B and O and was awarded Nobel Prize for Medicine for his work in the year 1930. We all know that the blood present in our body, it flows through our heart, arteries, veins and capillaries. Our blood has two parts, a cellular part and a non-cellular part. The cellular part constitutes of the blood platelets, the red blood cells and the white blood cells, whereas the non-cellular part, which is the liquid part of the blood in which the cellular part floats, that is known as the plasma. Karl Landsteiner, he discovered two things about the human blood. The first thing was that the red blood cells of the humans, they have a glycoprotein which is known as the antigens or the agglutinogens, or which are also known as the isoagglutinogens on their surfaces. And the second thing was that the blood plasma of humans have gamma globulin proteins which are known as the antibodies or the agglutinins. The two antigens discovered by Landsteiner, they are the antigen A and the antigen B, and these two we can represent by the letters capital A and capital B. The two antibodies found in the blood plasma, they can also be represented by the letters of the English alphabet. We can represent them also by capital A and capital B, or sometimes they are also represented by the small letters small a and small b. Now, based on the two antigens A and B and the two antibodies A and B, Landsteiner divided the human blood into four groups. Group A, which had the antigen A on its RBC surface and the antibody B in the blood plasma. Group B, which had the antigen B and the antibody A. Group AB, which had the antigen A and B but it had none of the antibodies. And the fourth group, Group O, which had none of the antigens but it had both the antibodies in its blood plasma. It was also found out that if the antigen A reacts with antibody A, then a chemical reaction begins and the blood begins to clump, or the process of agglutination of blood takes place. Same thing applies with antigen B and antibody B. So if these kind of blood transfusions take place where the mixing up of same kind of antigen and antibody takes place, then the clumping of the blood takes place and this results in the death of the person. So why earlier, for centuries many people used to die during blood transfusion, now the concept had become clear. So during the blood transfusion, proper compatibility of blood should be checked. Based on these antigen-antibody reactions, now it was decided that which blood groups can donate blood to whom and who can receive blood from whom. Blood group A, which has the antigen A on its RBC and antibody B in the plasma, it can donate blood to blood groups A and AB, but it can receive blood from blood group A and O only. Similarly, the blood group B with the antigen B and the antibody A, it can donate blood to blood group B and AB only, but it can receive blood from blood group B and O. The third blood group AB, which has both the antigens and none of the antibodies in the blood plasma, it can donate blood only to blood group AB, but it can receive blood from all the four groups, that is from A, B, AB and O. So as the blood group AB can receive blood from all the four blood groups, it is known as the universal recipient. The fourth group, that is O, which has none of the antigens but has both the antibodies, it can donate blood to all the four groups, that is A, B, AB and O, but it can itself receive blood only from O group. So as it can donate blood to all the four groups, now O is known as the universal donor. By the year 1940, Landsteiner, along with another scientist by the name of Wiener, he discovered one more factor present on the membrane of the red blood cells. Now these factors, they were the proteins which were for the first time detected in the blood of the rhesus monkey. So this factor came to be known as the rhesus factor, or this is also known as the rhesus antigen system. So apart from the ABO system, now the Rh factor also started playing a very important role. Now this Rh factor, it may be present on the RBC membrane or it may be absent. Now depending upon the presence and absence, a person can become Rh positive, that is the Rh factors are present, or the person can become Rh negative, that is the Rh factors are absent on his RBC. People with the presence of Rh factors on their RBC, they are represented Rh positive like this, and the people who have no Rh proteins on their RBC, they are represented as Rh negative as shown here. So now based on this Rh positive and Rh negative, now the blood group can fall into 8 categories. What are they? It can be A positive, that is the rhesus factor present, A negative, that is the rhesus factor absent, B positive, B negative, AB positive, AB negative, O positive and O negative. Now this Rh factor also plays a very important role during blood transfusions, and proper compatibility of the blood has to be checked before donating a blood to a particular person. Now let's quickly check the genotype and phenotype of the people with the presence or absence of the Rh factor. The genotype can fall in 3 different categories. Any character in the body is controlled by a pair of genes, we already know. So the presence of Rh factor is also controlled by a pair of gene. This is a dominant gene and Rh factor is a dominant character. So we represent the gene by the letter capital R. R is for rhesus. So first genotype, which can be dominant gene, both written in capital letters, and the person is Rh positive. This first genotype was homozygous dominant. The second genotype is heterozygous dominant, where one dominant gene and one recessive gene is present, but obviously the phenotype becomes Rh positive because of this dominant gene. The third category, where both the genes are recessive. So these genes, now in the absence of a dominant gene for rhesus factor, get a chance to represent themselves and the person becomes Rh negative. That means this person does not have the Rh factor on the membrane of the RBC. Let's talk about the blood compatibility here. Rh positive can give blood to Rh positive. Rh negative can give blood to Rh positive. But Rh positive cannot give blood to Rh negative. So this factor has to be taken care during blood transfusions. Now based on the ABO system and the Rh positive and negative system, we have here all the 8 categories of blood. Now let's check the compatibility of the blood types. A positive can give blood to A positive and AB positive only, but it can receive blood from A positive, A negative, O positive and O negative. B positive can give blood to B positive and AB positive, but it can receive blood from B positive, B negative, O positive and O negative. AB positive, it can give blood only to AB positive, but it can receive blood from everyone. Everyone means all these 8 categories of blood group people, they can donate blood to AB positive. O positive can give blood to O positive, A positive, B positive and AB positive. Earlier we had discussed that O positive is the universal donor, but now we know that O positive can give blood only to the positive group of bloods, but it can receive blood from O positive and O negative. Now we come to the negative blood groups. A negative, as we can see here, it can give blood to A positive, A negative, AB positive, AB negative, but it can receive blood only from the negative groups, A negative and O negative only. B negative can give blood to B positive, B negative, AB positive, AB negative, but it can also receive blood only from B negative and O negative. AB negative can give blood to AB positive, AB negative, but it being the universal recipient, AB being the universal recipient, this AB negative cannot receive blood from positive groups. It can receive only from AB negative, A negative, B negative and O negative. And finally we come to O negative. This blood group, it can give blood to everyone. All these 8 category of people, they can receive blood from O negative, but it can receive blood from O negative only. The blood group system of ABO provides a very good example of codominance. We saw in the case of rhesus factor students that if any one of the gene is dominant, then the factor has to be expressed. That person becomes Rh positive, but not in the case of these antigens. If the gene for antigen A is present in the cells of the person, then the person's blood group becomes A. If the cells contain the gene for antigen B, then the person's blood group becomes blood group B. But in the case where the cells of a particular person contain the gene for both these antigens are produced on the surface of the RBC and both these are expressed, and the person's blood group become AB. Because none of these genes are recessive, no one is able to suppress each other. In this figure we can find the percentage of the different blood groups found in the human population. We can see here that AB negative with 1% is the rarest blood group found, and O positive with 38%, it is the most widely occurring blood group found in the human population. The presence of antigen on the surface of RBC is a character present in our bodies, and this character is controlled by genes. We have studied that any character in our body is controlled by genes, and every gene has 2 alleles. Alternative alleles can be there, but in this case of the ABO blood group system, we find that it is controlled by 3 different alleles, and this is also known as the multiple alleles. The antigens are also known as isoagglutinogens, and so the designation of capital I, it stands for the genes. So the gene for blood group ABO has 3 alleles, and it is represented by IO, IA and IB. Out of these 3 alleles, IA and IB are dominant and IO is the recessive gene. This IO is the recessive gene or the recessive allele, so it can also be represented by small I. Now, when we compare these 3 alleles, IA and IB, they are dominant to IO. But IA and IB, they are co-dominant to each other. That means these two can dominate this particular gene or allele IO, but IA and IB, they cannot dominate each other. So both of them are expressed. Now out of these 3, the small I or IO, it produces no surface antigens, but IA produces the antigen A and IB produces the antigen B. Based on the above 3 alleles, the following types of genotypes and phenotypes are found in the human population. If a person's blood group is O, then his genes both are recessive alleles for his blood group. So it can be represented in IO, or both small I can be used here. None of the antigens are produced and the nature of the genes is recessive. If the blood group is A, then the genotype can be of 2 types. It can be homozygous dominant, both dominant genes IA, or it can be heterozygous dominant, one dominant gene IA and one recessive gene IO. The antigen produced is A and the genes are dominant. If the blood group is B, then again the condition can be both homozygous dominant or it can be heterozygous dominant. The antigen produced is B, the nature dominant. But if the blood group is AB, then only one genotype is possible, both dominant, one dominant gene for A, one dominant gene for B. The antigens produced are both A and B and the nature of the genes is co-dominant. Many times we are curious to know about what should be the blood group of children born to a particular kind of blood group of the parents. Okay, so few of the combinations we are discussing here. If one of the parents is having O blood group and the other is having A blood group. O, obviously both the genes are recessive, and if the A group parent, that particular parent is homozygous dominant for A, that means both the genes are dominant, then the possible genotype of their child, any child who is born to them, will always be one recessive gene and one dominant gene for A, and the blood group of all their children will be A only. Second example, again we take the blood group O and A. The genotype is recessive for the O group, but if this second parent with A blood group, if this parent is heterozygous dominant, that means one gene is dominant, the other one is recessive, then two combination of genotypes are possible in their children. One of the child may be having one recessive gene and one dominant gene for A antigen, and the blood group of that particular child will become A. But if the combination of gene becomes I0, one recessive from one parent and another recessive from the other parent, then the blood group of this particular child becomes O. That means two combinations are possible for their children. Third example, if one of the parent is having O blood group and the other is having B, that too homozygous dominant for B, again all their children born will have this particular genotype and all the children born to them will have B blood group only. Fourth example, if one of the parent is having O blood group and the other is having B, that too heterozygous dominant for a B, then again there are two different kind of combinations of blood group present in their children. Either their child may be born with B blood group or it may become born with O blood group. In the fifth example and other three, we will be discussing about the combination of blood group A and B of two different parents. One of the parents is A, the other is B. So for A, this parent is homozygous dominant for A and this parent is homozygous dominant for B. So always the combination of genotype or gene in their children will be one dominant gene from one parent and one more dominant gene from the other parent. And all their children born will have the blood group AB only. In the next combination, where the parent with blood group A is heterozygous dominant and parent with blood group B is homozygous dominant, then we can see here that two different combinations are possible in their children. One child can be born with both the dominant gene with the blood group AB, or a child can be born with one recessive gene from one parent and one dominant gene for another parent, and in the absence of a dominant gene, the blood group of this child becomes B. In the next combination, the parent with blood group A is homozygous dominant and the parent with blood group B is heterozygous dominant. So again two possible combinations are possible and two possible blood groups can be there in their children. And finally we come to another combination of AB where both the parents are heterozygous dominant. So this particular parent with A blood group, this parent is heterozygous for A and this one is heterozygous for B. So we can see here that four possible combinations of genes are there for their children. If the combination becomes one dominant gene from here and another from here, the child's blood group becomes AB. If the combination becomes one recessive gene from A group parent and dominant gene from B group parent, then the child's blood group becomes B. In the third combination, if this becomes a dominant gene from A group parent and a recessive gene from the B group parent, then the blood group becomes A. And finally, if a child receives the recessive gene from both the parent, then the blood group of this child becomes O. Similarly, some other combination of parents are also possible, like both the parents might have the blood groups A, one might be homozygous, the other might be heterozygous, both might be homozygous, both might be heterozygous. Similarly, the parents' blood group can be B and heterozygous or homozygous. So based on that, try to find out what could be the combination of genes in their children and what are the possible blood groups which can be found for these parents. And finally, let's discuss about two very unique blood groups. One of them is the golden blood group. This is incredibly rare blood type and it is found only in 1/6 million people around the world. It has complete absence of any of the Rh antigen or Rh proteins in its blood, and hence it is also known as Rh null blood group. Here it is also interesting to know that there are about 49 different Rh proteins which are found or which have been detected in the human RBC, of which RhD is the most significant one, and it is actually the RhD or the rhesus D antigen which causes most of the complications in the transfusions. But all these 49 Rh proteins, they are absent in the golden blood type. Because the golden blood is very rare, so donations are also very rare and valuable, because it is very difficult to find a donor. So these kind of people, they are usually asked to donate for themselves. They donate blood and they preserve it for themselves if at all they need it in their future. Another unique blood group type is the Bombay blood group, which is also known as the HH blood group or the OH blood group, and this blood group phenotype was first discovered in Bombay by Dr. Y.M. Bhende in the year 1952. Just like the blood group O, the Bombay blood group also lacks the antigens A and B. But in addition, this Bombay blood group also lacks a third antigen known as the H antigen from its RBC. This H antigen is found in all the other blood groups including O. So a blood transfusion between Bombay blood group and any other blood groups is not possible. HH or the OH blood group or the Bombay blood group is very rare, and only 1 out of 10,000 individuals in India possess this blood group. And if any blood transfusions are needed, the person has to contact another person having the same blood group, then only the blood transfusions are possible. Hope this video was useful and informative for all of you. For any kind of communication, please write in the comment box. Thank you all.